So I'm going to introduce George Hruby to you. He is the Executive Director of the Collaborative Center for Literacy Development and an Associate Research Professor of Literacy Education in the Department of Curriculum Instruction at the University of Kentucky. Right?
That's correct.
Okay, because there's an abbreviation of the UK here. That is,
oh, yeah.
Dice on Kentucky. Anyway, so before being at Kentucky, he was at Utah State University, where he taught professional development courses in learning theory, reading psychology, literacy instruction methods. In addition to a PhD in reading education, he has a BA in English from Syracuse and an MBA in language education from the University of Georgia. So there's some connections there. After spending several years in the workplace, he taught developmental college reading and worked with elementary students in a reading clinic. So his scholarship now involves critical synthesis of interdisciplinary research relatable to literacy education, most notably educational neuroscience. And that being said, here's George.
Thank you. Thank you. And thank you all for being here. It's a great honor to be here, and I'm proud of you for braving the weather. It is, but you're probably all used to this, right? On again, off again, winter. Well, I think it's picture postcard perfect, and it will certainly be memorable. I haven't seen a thing of the campus, but it's beautiful. Hopefully I'll see some more of it tomorrow. I'm not quite sure. Let me do a straw poll here. How many of you are highly knowledgeable about the neurosciences? How many of you are fairly knowledgeable about cognitive psych-oriented reading instruction or reading research? Okay. I'm going to I want to review some of the, give you an overview of the serious research. This is not a brain-based education presentation. This is not a pop psych, PD. This is a serious review of the research base, but I'm going to take it at a general enough level that it will give you, I hope, some themes that you can relate to what you know about literacy instruction. I'm going to try and relate it to literacy instruction as well. But you need to understand up front that neuroscientists do not study teaching and learning in classrooms. They study the brain. So if you want to know something research-based about teaching and learning in classrooms, you have to look at the research on teaching and learning in classrooms. And indeed, neuroscientists who, or cognitive psychologists for that matter, who would have something to say about reading instruction ought to be looking at that research as well, and they don't always do so. I am not going to review the research on pattern, metaphor, and narrative, although there is such research in the neurosciences, but it's far too formative to say anything of much interest, and it's a very higher order realm of analysis. The narrative I'm talking about here are the narratives that teachers and teacher educators rely upon to make sense of what they do and why they do it. Scholars, researchers have theory, and there's a very formal sense of what theory is. And the man and woman on the street have opinions. But somebody who's a practiced professional has far more than opinion, but not quite theory. So I'm going to call that a narrative. And narratives are organized and made concise and coherent by virtue of a central organizing operative metaphor. If you know the work of George Lakoff and Mark Johnson, you know what I'm talking about there, so I won't belabor that. And the metaphor that I want to offer you here is the metaphor of pattern. Because that is what the brain at a neurobiological level is all about, not information processing. Information processing is another fine metaphor for the brain that is very popular in the media. It's overdone, actually. And so I want to try and share with you an alternative way to think about it that I think is perhaps more useful. So a few preliminaries and caveats, and then share some insights from the cognitive neuroscience work, and then I want to briefly talk about some work from the social and affective neurosciences, and a little bit of developmental neuroscience or developmental science. That will infuse the presentation, and then I'll be out of time somewhere around then. So you'll have to ask me about theoretical narratives and literacy. But I'm published on that as well. I should say I'm not a neuroscientist. My PhD is in reading education. But I have formally studied that at the doctoral level. When I was getting my PhD over in the psychology department, and I have been a deep reader in it ever since. And I've tried to contribute to the field as a former president, program chair of the Neuroscience Education SIG at AERA and so forth and so on. Cognitive neuroscience is an interesting field. It's one part cognitive psychology, one part neuroscience. And as some neurobiologists have noted, just because you use neurobiological methods in research, it doesn't mean that the research is neurobiological in nature. And a lot of cognitive neuroscience is of that sort. It's taking the modular categories of mental process that have been hypothesized by cognitive psychologists and tries to map it onto the brain from sort of a whole brain architectonic perspective. Unfortunately, that is not how the brain works at that level. It works at a much deeper level. I want to share some of that with you. And so it's a little touch and go. And I want to point out that if you go to the Society for Neuroscience, which is like the AERA of the neurosciences, which is about 5 times as big and really hard to get into, but I've had the good fortune to get in and present at a couple of their national conferences. If you look at that, there's 10 areas in that society, and only one of them is about cognition, and most of the work in there is about neurobiological correlates to putative cognitive processes. They're not proving anything about cognitive theory, okay? It's used as a structure, as a rubric by which to make sense of their work. Now, like any area, cognitive psychologists have their metaphors too, and their central metaphor is that the mind works like an information processing system, like some sort of a software design. And this notion has become quite popular in the media, and you often hear about brains as information processors. Neurons are wires, action potentials are signals. What's happened here is that the nomenclature, the loose metaphors of cognitive psychology have colonized popular understanding of neuroscience. And neuroscientists hate this, most of them. Or at the very least, they're agnostic about this science. This is an example of what I'm talking about. This is I don't know if you can see this so well in the light here, but this is a very beautiful artistic representation modeled with clay, I guess, of what's supposed to be a neuron. And the only problem is it's not really a neuron. I know that from the shape. It's an astrocyte, which is a glial cell. Most of the cells in your brain are glial cells, and they don't signal, and they certainly don't light up. And neither do neurons, for that matter. Okay, your brain does not light up when you're thinking, even if you're a smoker. And you're not left brain or right brain. You don't just use 10% of your brain or any of this kind of stuff. So. But this image, this kind of thing is regularly imposed. This goes back to the dawn of the information processing era. This is the IBM ENIAC in the 1940s. It almost does kind of look like a brain with all these wires, but by the 1950s, early 60s, it's looking like this. This office probably has about 1/10 the processing power of your smartphone. But the work on software design at this time, Newell and Simon and folks like that, combined with experimental psychology, work by people like George Miller, and psycholinguistics, work by people like Noam Chomsky, came together to spawn what we call the cognitive revolution. And these tools have made their way into our offices, into our homes, into our classrooms, into our pockets, and into our heads, if you believe the media. Most recently, I know if you've seen this banner ad, it's really hauntingly creepy. They photoshopped all these faces so the eyes are just a little too big, too far apart, and angled to make you think of E.T. Cognitive inventory is here. This is the IBM Watson thing. What are they inventorying? The contents of your head. They want to know what kind of choices you make when you're on the internet. They're going to collect all that information. The kind of simplistic dichotomies that we like to celebrate in the media, right? Good versus evil, nobility versus savagery, right? And of course, it's all because of your brain. And from what I know about the brain, Mahatma Gandhi was quite the manipulator, and Adolf Hitler was a real good listener. I mean, that's what they're saying. But anyhow, originally, cognitive psychologists didn't care very much about the brain. They said it doesn't matter, it's the all-purpose hardware. It's the software design that matters. And like software designers at the time, they used flow charts. And the idea was that certain sub-processes were modular in nature, and they didn't bleed over into other sub-processes. So you could just, use factor analysis to be very precise about the effects. And then along came brain imaging in the late 80s, early 90s. And suddenly the modularity was there for all to see. Oh, well that must be that sub-process we were talking about. Look how nice and neat it is. The problem is, the modularity here is purely an epiphenomenon of the technology and the methodology being used. Okay? Nonetheless, for over a decade, there was an ongoing debate about is the brain modular or distributed? There's no question now that the brain is distributed. Let me explain a little bit about this imagery, because I understand now you're going to start using some of this stuff, so it's great. There's no such thing as a control state for the brain or you'd be dead. So you compare an experimental target with a very close comparison, so that something just, very specific variable is being signaled out. And then you subtract one from the other and you get a difference. Looks good. Then you do it again, and again, and again, because every time you do it, this is with one subject now, you're going to get a different result. Now you average it all together. Okay? Now you do the same with your next subject, and your next subject, then you average all of them together. This is the reason why so many neuroscience studies only have like 7 to 12 subjects. You think, wait a minute, if I can't get away with the reliability of a classroom, how can they get away with 7 subjects? Well, they thought that they could do this because they're doing multiple measures of each subject, but the problem is they're trying to generalize from the number of subjects to the general population, so that doesn't hold up, right? And it's only been in the past 3 years that anybody's actually published a critique about this within the neurosciences. But there's been a lot of other critiques in neurosciences. This is one of my favorites. Walford and Bennett have written serious papers, but this is a study they did with a salmon, and they found that there's a particular area in the brain of the salmon that responds to highly emotional human situations, as indicated by photographs that they showed the salmon. And the most remarkable thing about this is that the salmon is dead. They got it at the fishmonger, and it was fresh, but dead. And what this is about, of course, is false positives. And the way that this stuff is calculated has been troublesome. But a lot of the issue is this is a domain that has grown exponentially over the past few decades, and it is, in terms of average age, disproportionately young. Very bright, very sharp, young scholars, but they don't have a lot of seasoning in terms of research design or that sense of fallibility that comes with middle age and experience. So here is, this is the most promising thing, I think, in terms of critique. And this is from Evelina Frigorenko and her colleagues, stressing the importance of getting away from these averaged images and then trying to use this as a norm for all to be compared against, and realizing that the individual images are quite distinct. Yes, you can localize them within general areas, but look at how different these are. And this is from looking at a whole brain perspective, and we're talking about neurons you can't see with the naked eye, right? Or even a magnifying glass. So that is a world of difference. Then there's what's going on underneath the cerebral cortex. And all of this we now know, thanks to whole brain imaging, is also involved even in things like reading. We're going to get to reading, I promise. And there's diffusion tensor imaging now allows us to visualize white matter tracts, which are neurons that are sufficiently mature from lots of practiced use that they have grown large and strong from all that glucose and oxygen and nutrients they've taken up, because the neurons are not wires, they're living cells, and they need to eat and breathe just like you do. And the more they do that, the stronger they get, and they develop a fatty sheath called myelin, which makes them look white. And so it's called white matter as opposed to gray matter, which is more immature, more plastic and pliable. But this allows us to see where the most use has and in what direction it's flowing. And interestingly, a lot of the direction of processes such as in reading are top-down. That is to say, they're coming from the executive areas of the brain to the perceptual areas to direct perception. And some of you may remember this if you're familiar with the reading wars debates and so on. Right? Well, you'll see and for those of you who are like newer graduate students, always bottom up, top down. Sounds like New Orleans and Mardi Gras, right? Bottoms up, tops down. Well, this is then there are the neurons themselves. These are now we'll get into this. And then there's the chemistry, and this is just the basic chemistry of neurons and glial cells. And then there's the chemistry of the synapses. These are not on-off switches in a binary array. Right? This is not what's going on here. And this is from Carter and Shees, a couple of Stanford scholar neuroscientists, their methods text. Half of the chapters in that text are about genes and biomarkers, all right? And these are some of the predominant means of studying neural phenomena. There's only one chapter devoted to whole brain imaging. So I want you to get the media bias out of your mind and put this in a scientific perspective. And use your brain when you think about it. Right? Okay. Well, George, if the brain is not an information processor, what is it? I'm going to suggest that you should think of it as a pattern sifter, learner, predictor, and associator. All right, now that's not the whole story. But at the level of neural organization, because of the architectonic nature of cerebral tissue, where you have layers upon layers of networks that are then columnarily organized by pyramidal cells like elevator shafts in an office building, the ability to track patterns is literal. It is not about symbolic representation. There are no symbols in an action potential or in a neuron or anywhere else at that level that anyone has been able to find. We certainly know about frequencies of firing rate of action potentials, and there's nothing there that takes on the pattern that you would associate with a code. Now, yes, we do have symbolic phenomena in consciousness and in our culture. There's no question about it. We're a symbolic species, as Terence Deacon says. But we're the only species that demonstrates that. When you look at a nematode with all of 302 neurons, you can't tell me it lives and does what it does because it's processing information. Information in the technical sense of being a symbolically represented quantity. It's not what's going on. We use symbols to make sense of the world scientifically, and neuroscientists must needs do that. It does not follow that that which they are studying is symbolic in nature. That's an anthropomorphic fallacy. You understand what's going on? So, or you would call that a Platonic fallacy. Right? So, your eye, the retina of your eye, has 3 levels of neuron. The lens in your eye focuses the image, the light energy reflected from the world around you, lands on that retina, and in those 3 levels, in cluster surround arrays, the neurons calculate the difference between light and dark, boundary, and color. And that's all done before it gets back to the occipital lobe to process vision in the brain. So I'm saying and when it gets to the visual area, there are 7 layers there, but the first 2, VO1 and VO2, spatiotopically re-represent what was on the retina. Now why? There's no little homunculus in there to look at it. That architecture is crucial for what the brain does next. Eventually it gets to a level where you're going to be able to associate words with these patterns. And now we're cooking with symbols, okay? But how we get the symbolic from the non-symbolic world, we do not know. How we get consciousness from the non-conscious world, we do not know. And that is not a minor problem for educators. Because if your children aren't consciously able to recall what you've been teaching them on a test, you're going to say that they didn't learn it, okay? So, and consciousness is a mystery. Don't believe some of this brain-based stuff. We now know how the brain reads. We now know how the brain learns. No, we haven't conquered that symbol conscious, the hard problem in philosophy of mind, as it's called. Patterns are learned, reinforced, and fine-tuned from repeated and varied encounters. And automatic processes are the ability to tap those patterns more efficiently. When I mean efficiently, I mean in terms of biological resource dedication. As a neural network gets strong, as it becomes myelinated, it needs less glucose and oxygen to function. That means there's more glucose and oxygen in the bloodstream to do something else. That is how working memory expands. That is how processing speed expands. There's no space in your brain for working memory. It's the amount of glucose and oxygen you have in your bloodstream and your brain. You can only hold so much. And if it's being used in one place, it's not available somewhere else. That's why it's measured number 7 plus or minus 2. It's not a CPU. And that also then means that patterns must be overlearned to be anticipated. And that's what we do when we teach children to read every letter and every word. We're teaching them to overlearn the orthographic probabilities of certain letter sequences, not the spelling of every single word. I'm going to show you the neuroscience of that. All right, let's make sure we're on the same page. You're looking on the left side of the brain, like if you were looking through me right now, right? Frontal lobe, where executive processes tend to be. Temporal lobe, word meaning, we're looking at the left side, so this is a lot of language, tends to be in right-handed males at least. Left lateralized, 60% of left-handed males as well, about 50% of women. Women are much more bilateral. They use a lot more of their brain when they communicate with you. Already knew that, right? Sorry. And then there's a occipital lobe for visual processing. The parietal lobe is the least well understood, but it seems to be a staging area for new information to figure out the patterns. And any developmental, whether it's the sensory motor stage, the concrete operation stage, any of your Piagetian stages, it starts here first, and then later on it centralizes somewhere else. So it makes good sense that this area, which is the area that tends to get dedicated for letter-sound relationship, once that is learned, it winds up disappearing and being activated down here. I'm going to just pass. This is an old-fashioned view, but I'll just pass. These are the areas that we know are important in language comprehension today. Last year, Hickok and Small came out with the wonderful handbook, Neurobiology of Language, about 1,200 pages long. It's wonderful, fascinating reading. And I'll talk about some of these areas briefly. Here's your brain, about 3 pounds of tissue, wet, gooey, bloody, washed it off here. If you divide it sagittally, which means right down the middle, you can see that the midbrain and brainstem here is a lot going on underneath. And all of this is every bit as important as anything that's going up here in the higher order areas. If you were to separate this anatomically instead of sagittally, you would see the fibers, and you can see this here, where they scallop down. All of them come to the corpus callosum, which connects the two halves of the brain. That's why you're not right brain, left brain, I promise. And again, diffusion tensor imaging allows us to see these white matter tracts. And it allows us to see where they're going. This is looking at the front of the brain, but the gray stuff's been pulled apart except for the spine in the middle. And it's pretty fascinating stuff. Now, you know the simple model of reading? You know this, right? Okay. Goff and Tunmer, right? 87. And it's reading is the product of decoding and language comprehension, not the sum, but nonetheless, we're going to use a sort of a subtractive method because we're looking at neuroscience. You know the decoding sub-processes, you know the reading comprehension sub-processes, I don't have to tell you about that. This is a simplified chart. So if you look at what is going on in the brain when you do all of this, and then instead of reading, you have subjects listen to that language, and that would be what's going on here, and you subtract the two, what you have left over ought to be this. And that's the idea. Here's the reading comprehension. Here's the listening comprehension. You subtract one from the other. I already see Dana nodding his head no, and he's right. But that's how it got started. Well, I'll get to the conclusion of the story. Here are the 3 areas for decoding that we now know are really very important. The first area that was discovered, this is around 15, well not discovered, but slowly emerged, is the temporal parietal cortex here in the back right. There's a lot going on in here. Letter sound matching up here, sound blending here, and down here is Wernicke's area where the auditory word form is recognized. So you have cat, ding, right, that's right. And instantly the temporal lobe will be activated for meaning. And so will the frontal lobe, we'll get that in a second. The blue area is the occipital temporal cortex, the visual word form area, and it's a different color because it has nothing to do with phonology. Some people don't like the sound of that. I know there were theorists 20 years ago who said that even adult readers who are literate are actually processing every letter and every word. They just do it so fast they don't know they're doing it. That's not true. And then finally, there's the inferior frontal gyrus, and this area is Brokaw's area. It's the area for language generation. It's the language for syntax processing, for some semantic processing, but also for phonological processing. This is the area, or the premotor cortex here, happens to coincide with what in macaque monkeys are called mirror neurons. Neurons that fire not only when you do something, or the monkey does something, but when you or the monkey sees a conspecific doing the same thing. You fire up those areas as if you were doing it, so that that's how you're comprehending it. This is fascinating that phonological processing would be going on here during decoding, because what this suggests is what Johannes Peter Müller, back in the early 19th century, suggested. He was a German physiologist, and he suggested, goes, you know, when you measure the sonic qualities of language sounds, particularly when you watch vowels descend into schwa sounds, they're too close to be able to distinguish accurately just by the sound alone. And when you look at the differences between the way people speak, given accents, given dialects, given age and gender and so forth, there's no way that just on the sound alone that you could understand language. And he proposed that the way that we do it is that our brain activates the neural architecture for generating the sound when we hear it in order to determine what it is that you would be doing if you were making that sound, and that's how you identify the actual phonological category. Pretty smart. It turns out that that idea was taken up by Alexander Graham Bell, by the way, and used to help the deaf learn to read lips, because they're not really reading the lips, the mouth, the thorax, the lungs, and use that. And it actually, there seems to be a lot of potential, renewed potential at Philip, but you won't read that about Mueller if you go to his Wikipedia page, but Philip Lieberman at Harvard dug that out of the archives. Now, the reason why these two areas communicate so quickly is a white matter tract called the arcuate fasciculus. Okay? Now again, remember, white matter is something that develops because of use and experience within an environment that requires it, okay? It's not, the propensity to be able to develop it is innate, but the development itself is not. Remember that. It's always in response to the environment. Here's the study of kindergarten students. And they're imaging, and again, they're using a signal threshold that makes it look modular, but it's actually a bundle of fibers. And they looked at these kindergartners and wanted to, and gave them a test of basically their linguistic skills as a predictor of future possible reading problems. And unfortunately, in the title of this piece, they refer to that as dyslexia. It's, you know, and they're not doing a longitudinal study. I get all willy about this, but anyhow. But what they found with those students who have better language skills, lo and behold, have a much larger and more pronounced arcuate fasciculus. So the experience is actually being manifested in the structure of the brain, right? Learning changes your brain, there's no doubt about that. Here's another view, and here it's blue. Your arcuate fasciculus is blue. Here's Brocau's area, and over here is Bernanke's area. Over here, this green is the visual word form area for sight word reading. Note, it is not connected to the arcuate fasciculus. It is connected to the fusiform fasciculus, the yellow-orange tract you see here, which runs from the occipital lobe for vision, and particularly that VO1, VO2 were patterns, and the inferior parietal for letter identification, at least as recognizing shapes as letters as a category, straight down into the temporal lobe for meaning. Subsequently, you will see auditory word form activation, but it's sort of like subvocalization as a sort of a subconscious recheck. Now, let's see this in action. You all remember, not some of you don't, Phil Goff's 1 second of reading. This is what goes on in the mind during 1 second when you look at a word, all right? And it's a rather clumsy model, but it was a start, and a lot more elegant models would come. But let's look at how it works in the brain. I hope you can see this okay. Can you see it okay? You'll see the activity, but we have this as bright as we can. The gray area is a model. This is from the Max Planck Institute, and this is based on EEG studies that have been sort of meta-analyzed. And the temporal lobe is down here. It's been pulled back so you can see what's happening on the top. And behind it, the occipital lobe has been pulled out so you can see it from the side as well. The green are sensory processing, visual, auditory, articulation. The blue are your decoding processes, letter sounding, auditory word form, visual word form. And then your red are your comprehension processes, word meaning, syntax, semantics, and 2 little asterisks that you probably can't see over here for emotional processing. emotional association at the anterior lobe of the temporal lobe, and then the orbital frontal cortex for emotional self-regulation. Okay? So let's watch and see what happens when you read a word. It won't even take a second. It'll take about 2/3 of a second. Got that? Oh, you missed it. All right. Let me stretch it out so it takes 10 seconds. More likely to see. What do you think is going to happen? Anybody know? What would start first? What's the first thing your brain would do when at 1 second you look at a word? Visual. Visual, okay. Yeah, and then what? Well, then it depends on what theory of reading you believe in, right? Okay, so let's find out. Let's try that again. Keep your eye on your favorite spot. Did you get that? What's happening here is it's starting from the back. Visual processing is run. These are adult readers, all right, and they're reading sight words. So you're not seeing anything going on up here. You are seeing it going on down here at the visual word form area. It's running forward into here, and at the same time you're seeing the audio word form kick in. Then you're also seeing, of course, thanks to the art of the fasciculus, activity up here. Also syntax, EEG studies show that syntactic identification starts even earlier than word meanings in some cases. And you're also seeing an interesting amount of activity in the emotional processing areas. Let me do this one more time. And then lastly, there's a recheck. So you're visually grabbing some letters, whether it's as a sequence or as a cluster, and you're shooting right down and predicting what it ought to be, and you know, right, on the basis of what cues you have. And then after you think you've got that, you go back and revisualize and recheck. How about if we did 1 second of listening, what would you see? Auditory, no visual processing, right? No visual word form.
Well, you made me think that maybe there will.
Oh, I'm going to play gotcha? You think I'd do that to you?
I know you would.
Let's take a look. One thing you might notice, keeping your eye on the clock, it takes a lot longer to hear a word than to read it if you're an adult reader. That's one of the values of print text, of course. Also, look at how much activity is going on in the emotional areas. All right? One more time. Much more intense. It's the sound of a human voice. Yes? I'm sorry, I see a hand? No? No. Question? And again, these white matter tracks are there to make this happen so quickly. All right? Now, there's another bit of neural architecture here, a subcortical area right underneath the temporal lobe, and it's the hippocampus, where memories are formed and reconstructed during recall. Right at the end of it is the amygdala. Some of you may have occasionally read across this in the media. This is your fear monitoring organ, but it actually is involved in any kind of emotional tracking. And it's not by coincidence that it's right at the anterior temporal lobe where emotional associations are made, nor is it coincidental that it's attached to the end of the hippocampus, because, and by emotional I don't mean hyper-emotional, but everything that's meaningful has some emotional content, right? Kids who are bored out of their heads in a classroom don't learn very much. They remember that they were bored. But, it's well understood. You already know this, folks, right? This isn't an insight. Learning occurs most optimally when learners are intentionally engaged in what they take to be inherently meaningful activity. And that's why, but the meaningfulness is inherently visceral, right? It's embodied. Welcome to social neuroscience, affective neuroscience, and neuropsychiatry, okay? This studies the neurobiological correlates of interpersonal relation and socio-emotional response. So if you think neuroscience is only for the cognitive folk, not for the socioculturalist, well, some socioculturists don't go do this with a 10-foot pole, but there is potential here, particularly in studies of close classroom learning and dyadic and small group behavior. Here's some brain imaging of a young mother, and she is being shown a stack of pictures of babies, right? Young mothers, babies, perfect match. She's looking at them, and she's going, oh, would you look at this one? Is this yours? Oh, this is beautiful. Look at those eyes. And then, stuck in the mix, is a picture of her own child. You take a smile. You can almost feel the oxytocin. And which of these sets of images do you think is the mom when she's looking at her child? The visual processing, this is looking top down on the brain, this is looking straight at her head. The visual processing areas are relatively the same, but what is different is anterior temporal lobe and orbital frontal cortex. All right? It's not just the mom who has that experience. Within weeks of birth, the infant and the mother or the caregiver are in a dyadic relationship where they are co-constructing the infant's emotional self-regulation. By 5 years of age, these areas will be highly developed in terms of white matter in a 5-year-old child. You're going to say, a 5-year-old child? What about those kids who have meltdowns at the grocery store? Well, you're talking about something else. You're talking about compliance to social protocol, right? The children, most children, in spite of the fact that they're still experimenting and testing their limits right up until they're about 30, will do have that, or should have that. And it's because, again, of this relationship here. This is crucially important for learning. Now, here's a question. What happens if they don't get that dyadic relationship? Now, no one would do that experiment, but it was done for us in nature with the Romanian orphans. Some of you may remember many years back, Romania is rather morally conservative country, and if you are pregnant out of wedlock, you go off, have the baby, and give it up for adoption. But unfortunately, it was a very poor country during the time of the Warsaw Pact, and so they had these huge facilities like big box stores, bumper to bumper with cribs, and a handful of care providers, barely enough time to run around, feed, and change the diapers, and so on. So these infants were neglected. When the wall came down, a lot of these children were adopted, but about a third of them could not be put up for adoption because they were so disabled, behaviorally, cognitively, no language skills, that they had to be institutionalized for the rest of their lives. Another third could be adopted, but they proved to need a lot of support. And then the other third seemed to be fine. But of course, we don't know which kids got there for how long, how old they were when they got there, and so forth. And and now they're in their 20s. But back when they were 13 to 15 years old, a group of Israeli scientists did some study, and this is in brain state. This is at rest. Okay, so it's not any particular function, just at rest because the brain is always active. Top down, here's the front of the head, here's the back of the head, and here are the anterior temporal lobes, okay? These are normal 13 to 15-year-old kids. Here are the Romanian orphans. It's as if they'd had lobotomies, all right? So if you don't get that kind of dyadic, co-constructed, emotional foundation, subsequent language development, and thus subsequent cognitive development cannot follow. This would seem to be the conclusion, all right? So, this is something, again, that you already know intuitively. I should also say this has something to do with equity and justice. The systems that allow us to respond to threat run throughout our entire body. The autonomic nervous system runs through the entire body. And the sympathetic system and the parasympathetic system work in tandem to respond to the environment. The sympathetic system is what we call the fight or flight response. Right? You see a tiger coming. What are you going to do? Your blood rushes to your arms and legs. It leaves your digestive tract. It leaves your cerebral cortex. Not the time to be philosophizing, okay? And the parasympathetic will kick in at the exact same time, but most of the parasympathetic signal goes through the bloodstream. It's a tie-in between the endocrine system and the nervous system. There's no such thing as a hormone-free thought, and there's no such thing as a neuron-free emotion. These two things work hand in glove. What happens with somebody who's been traumatized with chronic stress or severe stress is that the parasympathetic system habituates. You know, well, I guess that's the way it's supposed to be. The sympathetic system never does. And so the parasympathetic system not activating does not shut down the sympathetic system. And you get the sympathetic system on 24/7, even in the absence of external stressors. You may have had kids like this in your classroom. All right. Now, as you can see, it's a whole body phenomenon. What sorts of other effects are there? Well, high blood pressure, increased rate of stroke, increased rate of heart attack, increased rate of digestive disorders, ulcers, diabetes. I could go on. Depression, aggression, self-medicative behaviors, and cognitive impairment. And you can look at the demographics to see where we find people, I mean, people all over our society suffer from these things. But there are clusters that are worth noting. The cognitive impairment is neurologically demonstrated as well. Here are tissues from rats, thankfully, hippocampal tissue. These dots are the pyramidal cells that have been stained. Obviously, this is from an post-autopsy. The dark areas are dead pyramidal cells. These are a normal rat, at least as normal as you can be as a rat living in a laboratory. And here is a rat who's been chronically stressed. It's the neurotoxicity of testosterone, of adrenaline, the absence of serotonin and dopamine, and it does have a negative impact on cognitive performance. So if you think that anybody can just pull themselves up from the bootstraps, no matter what sort of environment they've been born into, when you know that spittle assays of non-combatant citizens in low SES urban environments in America have been analyzed, 40% of them suffer from the corticosteroid levels that would be associated with post-traumatic stress disorder. So while it's quite possible that if somebody who's as dim-witted as my brother-in-law, given enough time, could wind up poor, it doesn't mean that kids born into poverty are poor for that reason. If they are struggling in school, it is among other reasons, it's because our system literally assaults them neurologically. Which just reminds us that we are social beings. We need one another. But we're rather ambivalent about that sociology. So, I would remind you that when you think about meaning, which is an important component of comprehension, you have to think about the fact that it's a visceral, embodied state, literally, physiologically embodied, neurologically embodied. And at the neurological level, learning is about associative regularities which are identified as patterns across space and sequence, and most of these are chiefly subliminal. You've been a teacher, you taught second grade for 10 years, you got it down. What you're not aware of is most of what you're responding to, the regularities in your environment, you're not aware of. And then the principal says, could you do 4th grade next year? I want to change up the staff a bit. Sure, okay, I taught some of those kids 2 years ago. Totally off your game. Or you go to a school across town, second grade, still totally off your game. Or you're a teacher educator and you got your gate down at one university, you go to another university, totally, what the heck happened? I thought I'd figured this out. Most of the regularities that we respond to, it's not consciously directed. This is pre-conscious, subliminal sort of, but it is going on in the brain all the time. This is what brains evolved for, to allow us to functionally respond to our environments. And in the case of humans, those environments are social, linguistic, symbolic, and cultural. Cross-association allows for metaphor and analogy, and it's mirror neurons. This is an area that still needs a lot of investigation, but it's fascinating. And this does relate to our teaching. Phonics for decoding. As the neural illustration that I showed you would suggest, you don't want to make the mistake that whole language people made back in the 80s, or that the Gestalt psychologists made back in the 1930s, yes, you do have to teach kids to read every letter and every word so that they can overlearn the letter probabilities, okay? This is why before we had phonics, we were just as good teaching kids to read with spellers. C-A-T spells cat, okay? And there are other ways to do it, and it doesn't have to be a dismal experience. I'm not talking about rote drill, okay? But remember, there's that emotional part too. All right. Comprehension strategies, I'm going to argue this, but I don't have time. But Victoria and I can have an argument. In fact, we have had this argument. That strategies do not improve your comprehension. Strategies are protheses for developing skills which are about your improved comprehension. And you have to practice those darn things until you become fluent. It's like finger drills. Finger drills are horrible music, but they allow you to develop the musculature and the neurology to have the dexterity to play good music. And then text structure. You know, Ray and his colleagues at USU have done some work on this. Disciplinary learning, I'm going to argue. The reason that we don't get transfer across disciplines of basic skills is because the skills aren't generic. The skills are associative patterns within a disciplinary environment. Yeah, okay. So, I don't need to explain your own work to you. Let's see. Although I could. I don't have time for this. So, if you know about Uri Brenner's bioecological theory of human development, and this has been very popular, Linda Purcell Gates says we need this to pull together cognitive and sociocultural. Dave Pearson says we need this. I want to remind you that it's bioecological. He didn't call it ecological. And he said the same thing was going on inside. He just didn't have the science back in the 80s to explain that. The brain does not, this is 15 years of development in 4 seconds here. It does not develop equal potentially, that there's certain things develop before others, and that's why certain things are worth teaching kids at certain ages and not others. But every individual is going to be different. Well, I'm not going to get into this. My colleagues and I at Kentucky, University of Tennessee, and UNC Chapel Hill have been working on this for a year and a half, but it has nothing to do with neuroscience, but I just want to try to get that in. Anyway, it's in press, so what can I tell you? What I can tell you is what you already know, that there are these 2 general metaphor-grounded theoretical frames, cognitive psychological theory, that learning and literacy are symbolic representation of processing in the head, sociocultural theory positing that learning and literacy are socially mediated, culturally historical, the situated action and identity, called the crowd. Bioecological theory would posit learning and literacy as organically developed processes for attuning humans to their shared environments, including social and linguistic environments, and that's straight out of current developmental science. When we talk about developmental reading, we're always citing Vygotsky and Piaget. Great work, but how would you like it if a developmental psychologist told you he's researching reading and he grounds his work in the ideas of William S. Gray and Edmund Burke Huey from the beginning of the 20th century? Said this isn't serious, right? That was so. I mean, we could do with some updating here, and I think the neurosciences provide a fascinating window on that. So I think I've made my point. I won't overdo it. Have you ever seen this painting? This is the Valley of Wyoming. Did you know that Wyoming Territory was named after Wyoming Valley in Pennsylvania?
Yeah.
Yeah. But I bring this up because it's a naturalist idiom. And I think we could do with some neo-naturalist, neo-functionalist thinking about learning and change over time. It is to a large degree inherent in human beings to learn from their environment, but the outcome is external. And you know, when you look at these wonderful naturalistic paintings, there's always the human element, there's always nature, and there's always a great attention to detail, and yet the composition, the organization of it is so wonderful. I just think it's great, and I especially like how often they're reading a book. I think that's how they kept their subjects still while they painted. I love this one. This is Jenny Briscoe's Love's Last Dream. You know, she's looking down the lane with her bouquet. That's kind of the visceral, emotional part of the brain. And this guy's reading his book. He's got his finger picking. He's got his finger stuck in a page in the back, an index or something. It's strategic reading. All right. And the gal here is knitting, so you know, action in the real world. And then, of course, the kitten with the ball of yarn at the bottom, right, representing the fact that it is neurologically endowed organisms are meant to engage with and enjoy and learn. So I leave you with that. Thank you very much for your attention. Comments, thoughts, disputations, questions, ideas?
Oh, I'll start because what do I know?
Okay.
Yeah.
So I was interested at about 2 minutes ago, 3 minutes ago, the word transfer came up. And the whole game in school is based on transfer, that I learned it over there and I'm going to take it with me somewhere else, and it just mostly doesn't work. It's just too hard. It's too hard. So I'm to the point, I've been in this long time now, I'm to the point, I'm amazed things go as well as they go, given how little we really understand. So what would you say about transfer based on, because I started thinking about it when you used the word pattern, because I think that generally as psych people and learning theorists would say, get the pattern down, you just haul that pattern along, and voila, you're in advanced math or something. And there's no evidence that works very well for very many people.
Well, you know, you're right. The pattern has to be, to transfer, you've got to bring the pattern to a level of abstract representation and conscious thought, right? You're going to communicate it that way. But to actually understand that pattern, you have to have done it. This is something I feel with my teacher professional development work that I do. With my graduate courses. I do a lot of stuff in the field too. I don't do that there. But in my courses, if you're going to teach, and even I do this even if it's not a content area, literacy course, if you're going to teach strategies for improving comprehension to their students, they have got to do them first.
Yes.
Okay? And you've got to find texts that are at their instructional level of challenge so that they require that nobody needs strategies to read a 4th grade textbook, right? Or hopefully. And so you get them to do that, and at first they're like, oh, this is hard. Well, that's how it is for your kids, you know? So let's keep at it, and you work at it. But by golly, and I've been doing this for years now. I started doing this back when I was at Utah State. But by the last quarter of the semester, somebody, impromptu on their own volition, I don't prompt this, so it's like, you know what, Dr. Hruby, these things really work. I'm going to use this in my classroom. That's the idea. Thank you. But and they do work, but you have to understand how they work by you know, it's like being the method. You know, you have to do the method. You have to develop the patterns yourself that you can reflect upon it. And then that still doesn't make transfer easy, right?
Yeah, I was going to say that to me, it's the idea that as a learner, I'm going to look for patterns. It's just that in mathematics, the patterns you see are in numbers. In science, the patterns might be in reactions or some kind of end result from some chemical reaction, or looking in a microscope, you see patterns of movement. In social studies or history, the patterns are embedded in photographs and primary documents and things like that. And so you're trying to make sense of those patterns, and in each one of those disciplines, you use sort of a different way to make sense of that. You might graph some data, you might organize data in history and ask different questions. Who authored this piece? Where was this person situated? When was it authored? Was it, done during this war or this battle? Or was this looking back 50 years after this event? What else was going on, and how does this compare to the other data that I can see in other documents? And so to me, it really is very different depending on the discipline you're in.
Now, I want to caution that, as I said this at the beginning, pattern isn't everything. This is clearly what's going on at a neurological level and often generally subliminal. However, I think that when you get to the level of conscious, linguistically mediated warrant and logic and so on, that's got a logos to it all of its own, so that there's a lot more to it. But wonderful T-shirt while I was in the Chicago airport waiting to change planes. Fellow in front of me in line, and reading the back of his T-shirt, it says, "I'm a mechanic because your honor student who became an engineer couldn't get it right the first time." As academics, we privilege the abstract. We privilege that which can be reified and presumably at least intellectually transferred. But in the real world, including ourselves, most of what we do is about pragmatics, and a lot of what we do on automatic pilot, and we never think about, well, how do we ever learn to do that? I mean, if you want to have some fun, if you're right-handed, try brushing your teeth in the morning with your left hand, or vice versa. Just try breaking the pen. See how much fun it is. And then, you know. Thank you.
Thank you.